Thank you, everybody, for coming. It's always an honor to see an audience. This is, make sure you're in the right room. This is Climate Change, How Safe Are You? And this is the latest in a series of public talk series. So how many people came last year? Anybody? So we got about a third of the people who came last year. I think we should, special shout out to the BCIT students at the back who've been coming to these year after year. So this one's a bit special. They're all special, but this one's a bit special because we're actually going to run it into the fall as well. So we're having 5 talks now, one after the other, until the middle of March. And then we have at least 2 talks in the fall. One by the scientists who actually gave us the hockey stick graph, the famous graph of temperatures through time that's been called the hockey stick graph, and another by an economist who worked with Nicholas Stern on the Stern Report from the UK. And we're going to try to get a couple more. I want to get one on extreme weather. I want to get a talk on extreme weather, but we have to find somebody for that. And I also would like to get a talk on from an insurance company perspective, because I think that would also be very interesting. But we're still working on those. However, we do have 5 fantastic talks now, this wet spring. And I will introduce Dr. Lowenberger in a moment, but first I would like to invite the Vice President of Research of Simon Fraser University, Joy Johnson, to say a few words. And I think this is very important because this shows sort of the commitment that Simon Fraser has for this kind of engagement, that we get vice presidents to come and speak. So Joy.
Thank you very much. Thank you very much. I too want to welcome you to Simon Fraser University on behalf of the faculty, staff, and students. I want to really thank Professor Arne Moore for the role he's playing in organizing this series. I just think it's terrific, so thank you very much. It seems appropriate that this talk is being held on National Sweater Day, a day in which all Canadians are encouraged to put a sweater on, and I put mine on today, and turn down their own thermostat. I think that the issue of climate change is very pressing, and it's one that we all need to be thinking about. I want to begin by acknowledging that we're gathered on the unceded lands of the Coast Salish people. SFU's vision is to be a leading university and an engaged university defined by its dynamic integration of innovative education, cutting-edge research, and far-reaching community engagement. And I think that this lecture series is a prime example of this engagement. This lecture, as Arne has said, is part of a Planet Under Pressure series, and it began in 2011 and continues. Research now demonstrates that the continued functioning of the Earth system, and therefore the well-being of human civilization, is increasingly at risk. Pressure on our ecosystems are being exerted from many sides: increasing world pollution, unsafe levels of pollution of global air, water, and land, ecological changes, and increasing resource demands. We really do have to work together to explore solutions to this impending global crisis. Perhaps one of the greatest pressures of all is global climate change, the topic of this year's lecture series. Our global environment is currently in a period of unprecedented, rapid, and dramatic change with uncertain long-term consequences. SFU is addressing this pressure through 2 major initiatives. One is public engagement. Through lecture series such as this, where scientists draw on evidence to bring out the very real implications of their research and inform the discussions on evidence for climate change. The other is through research programs, where SFU faculty and students are exploring questions related to aspects of global climate change, including a long list such as ocean acidification, modeling of predicted changes, assessing risks locally such as sea level rise and on a larger scale, including impacts on coral reefs, loss of biodiversity, availability of groundwater, adaptations of salmon to warmer waters, economic incentives to curb greenhouse gas emissions, and public health and infectious diseases. I could go on. Last year's lecture series, Deep Time and You, examined global climate change through the lens of paleoecology. The lecture shows us how the study of past environments, plants, and animals can contribute insights into the long prehistory of natural changes on Earth and shed light on likely future consequences of ecosystem disruptions. Tonight, we begin the next series of lectures and discussions, Climate Change, How Safe Are You? Global climate change is threatening our security. We will hear from experts about the implications of changes for our food and water, our health and well-being, our international security, and even our pocketbooks. Embedded in these issues are pressing ethical dimensions. It is clear that we need to do more than understand issues. We need to lead and engage in evidence-informed, sustained action. Tonight is the first of 5 lectures. Please join us for all of them. It's now my pleasure to ask Dr. Arne Meurs, a professor of biodiversity in the Department of Biology at SFU, to introduce the speaker, and he will also be leading our discussion. Arne.
Thank you. All right, so you know we have 2 hours. Thank you very much. It's a joy for this. So it's, as you know, this runs for about till 9 o'clock, so we have about an hour for a leisurely presentation by our speaker, and then we have up to an hour for freewheeling discussion after that. So these are the people that are going to come the next 5 weeks. We're going to deal with disease, food, environmental health, security, and ethics, which I think will be very interesting. And then, as I said, we'll have more in the fall. But we're starting off with a very good colleague of mine, so a member of the Faculty of Science at Simon Fraser in the Department of Biology. And Carl Lowenberger did his first degree at Guelph. Then he actually did a Master's of Pest Management, I don't know how long ago, at Simon Fraser University, and Peter Belton was on his committee, and I think he's here in the audience. Am I right? Yes. And after his Master's in Pest Management, so he did his undergraduate was focused on entomology, then he did a Master's of Pest Management focused on parasites, and then he put them together and did a PhD at McGill University. Then he went to the States, as many of us do, and he did a postdoc and had a faculty position at the University of Wisconsin in Madison. And then, and this is one of these nice stories, the federal government about 15 years ago put together a program to lure Canadians back. It was called the Canadian Research Chairs Program. So the federal government would endow these chairs to bring Canadians back, Canadians of high caliber. And Carl Lowenberger got one of those, and we lured him back to Simon Fraser, and he's had that for about, you get one of those for, I think, for about 10 years. Now he's professor of parasitology, and he's also the associate dean of science. So he's a very busy man, but he agreed to tell us about climate change, disease, and your safety. So please join me in welcoming Carl Lowenberger.
Thank you, Arne. Is my mic on? Can you hear me? You know, 6 months ago when we put this group together, the steering committee, to try and find a bunch of speakers to get some topics, they said to me, Carl, would you give a talk? And I said, I really would rather not. And then we kept having people backing out. And they said, Carl, would you give a talk? I said, sure, it's 6 months away. It's not a problem. Now it's time to pay the piper, and here we are. So I hope I can convince you that there are some major and some very serious issues with climate change as we know it and as we predict will happen, and also where do we stand. This is a slide I actually hope we could send out to you and tell you that we are no longer having this seminar tonight because Pineapple Express came. I don't know how many of you got soaked and have your raincoats and your umbrellas at the back of the room waiting to dry out, but I was told, in fact, I was told very strongly that no, we were not canceling and we had to go on with the show. So I'll go through an outline of climate change, predictions and implications, and I'll try and show you that we actually don't know as much as we would like to know. I'll talk about some direct effects on disease transmission. We'll talk about some waterborne diseases and some problems that occur. Some vector-borne diseases, these are dear to my heart. As mentioned, Peter Belton was on my supervisory committee, and Peter's at the back of the room, and he is still one of the leading authorities on mosquitoes in Canada, if not North America. We'll look at human migration briefly and trying to understand what happens when we have climate change and we have strife and we have problems and people try to escape troubled areas and they move and they take with them the vectors or the parasites or the pathogens that cause disease in humans. We'll look at a few models. Great experience in my biology classes. Most biology students don't like models because there's too much math and too many numbers in it. But we'll try and look at some of the models that can predict what's going to happen. And the trouble with model prediction is that there's a great level of variability. And then we'll talk about are our fears justified? And because it's in the news so often, we'll talk about the situation, a little bit about Ebola and what's going on. And then we'll talk about what the heck can we do? And then we get your fun, you get the audience participation, and you can stretch or dance or ask questions and try not to make them too difficult. So before I go on talking about the disease transmission, I really want to say that climate change creates problems. That creates food and water insecurity, the quality of food, the availability of food, the availability of water, which means insecurity, which means migration of people, which people move to areas where there's massively increased density of people. And we know from good experience that as massive numbers of people get together, they transmit pathogens back and forth to each other. They have malnutrition, disease of transmission, overwhelmed healthcare systems, and we have a major problem. And all of those can be downstream of climate change. So we'll try and walk through the process of what are some of the parameters that lead to food insecurity and climate change and the effects upon people around the world. So climate change can be defined by different people. It's a significant and lasting change in the distribution of weather patterns over periods ranging from decades to millions of years. It can be as simple as a change in average weather condition or distribution of weather around an average condition. It can be changes in many aspects of weather, such as wind pattern, precipitation, and severe weather events we'll talk about, and far-reaching or unpredictable environmental, social, and economic consequences. So what does this really mean? It means that we have climate change, which is a persistent change or trend in mean conditions or current changes unprecedented in human history. And then we have climate variability, which is the day-to-day situation. Climate variability is a situation in which the weatherman can be wrong 75% of the time and still keep his job. Whereas these guys never lose their job simply because they say, well, we have to wait for another century or two before we can predict there, so keep me on and pay me. So this is short term, this is much longer term. So climate change can entail changes in mean or averages of what we see in the system, or it can be quite different in variance. So if we look at the dotted line up here, and this is what our standard, we'll use temperature in this case, standard temperature is the curve of cold weather, warm weather. And with climate change, we can simply shift that along. The curve stays exactly the same, which means we have less cold weather events or fewer cold weather events, more hot events, and more record hot weather events. So a simple shift of temperature up X number of degrees. We can also change the slope of that curve, push down the curve, essentially increasing the variance, even though the mean may actually not change whatsoever, but now you have more record cold weather events, more record hot weather events, and extremes. Okay. And then you can actually have this changing both ways. You can shift the curve and flatten out the curve, so we have fewer problems with the cold weather, and we have far more problems with hot weather and record hot weather. And we're in a situation in which we know this is occurring, and we have data that shows that some of the other scenarios are working as well. So I'm going to quickly go through a few graphs and try and set the stage for what we're going to talk about a little bit later. So we can have the average or the mean can stay steady all the time, and we have some trends, some measures around that mean, and the mean doesn't change and the extremes don't change, so everything's pretty stable. We can have the mean staying the same, and then we start to have increasing extremes around that mean, but the mean might actually stay the same. And we have this situation in many parts of the world where we look at the weather, we look at rainfall, and we actually have from some countries in South America where you measure the rainfall, the mean hasn't changed over many, many years. But now what we're seeing is events in which 6 months of rainfall fall in a week, and then the other 5 months of the rainy season are perfectly dry. But the mean doesn't change. Then we have an increasing mean with very little change in extremes, which we have, it could be temperature increase, it could be rainfall. And we can have what we don't want to have in terms of some of the things we're looking at, increases in our parameter, it could be temperature, and also massive extremes. And this is a situation we don't want to get. And lastly, we can get this mean no longer being a linear mean, but being exponential and going up, and includes all these different extremes of situation. And this is what my children would call crazy land. This is the crazy land position that we don't want to get to. And of course, because it's simply a mathematical model, we would never see that shape in real measurement of real data. Unless we get a situation where we look at changes in temperature going over centuries, and now we're getting the tip of the corner, and we're actually going up, starting to go up in some of that exponential phase. And once again, when we start to look at the background wobble, the influences, natural influences, we have carbon dioxide concentration, we have temperature in red, and it's hitting that crazy land concept. So in fact, the modelers tell you maybe it shouldn't be going up that because that's very unstable. We're going to have a whole bunch of problems. Some of the data we actually have say we have turned that corner and we're into the crazy land scenario. Okay. So then some of these crazy land scenarios lead us to extreme weather events. And as Arne said, we hope to have a whole talk about extreme weather events. These could be hurricanes, it could be floods, it could be other catastrophes. And these are simple extremes based directly on climate statistics. And they can be droughts, they can be floods, hurricanes, typhoons, cyclones. You can have modifications in rainfall or temperature, that you can have a month or 6 months rainfall in a single event. And it's the timing and duration of the extreme weather that affects disease. There will be a quiz on your way out. Climate change is not a simple, easy factor. So we've got climate change stuck in the middle here, but there are many driving forces which add to climate change. And the only thing I want you to take away from this slide is that there are direct effects, regional direct effects, and indirect effects upon climate change, which act through temperature and weather changes, through pathogens, through agricultural systems, and onto health effects. It's not an easy, simple, one-to-one relationship. Temperature goes up a degree, 10,000 more people die. It's rather complicated, and it's all meshed together, and we'll talk about later on the talk how we can pick this apart and why we have to have multi- and interdisciplinary studies to understand these different parameters. So let's take it down simple to a simple diarrheal disease. We're looking at high temperature, high humidity, high precipitation, which affects living conditions, water supply and sanitation, food sources and hygiene. Massive temperature, high precipitation, which means that the pathogens which cause these diseases survive better in the environment. They're not removed, which means there's more around for contamination of water sources and food sources, and then people can have a one-to-one contact and transmit these things. which means consumption of contaminated water goes up, consumption of contaminated food goes up, and ultimately you have incidence, number of new cases, and mortality. Simple diarrheal diseases. Why do we worry about it? Because conservatively, 1.5 million, some of the other data would say 2 million people die every year from simple diarrheal diseases, which are completely treatable and curable. 90% of these are children under 14 in developing countries. 88% of diarrheal diseases is attributed to unsafe water supply, inadequate sanitation and hygiene. And as I'm sure you've heard, 76.4% of all statistics are made up on the spot. But these are numbers drawn from the World Health Organization databases, and we have to believe that they're true. These are preventable diseases which are exacerbated by the problems of severe weather events. Imagine, if you will, a little community, massive rainfall goes into landfill, percolation, runoff into the aquifers, bringing water up the well to feed a community. You've got agricultural runoff. You can see all the circumstances in which anywhere there are pathogens or parasites in the system, they can get into our water supply, into our food supply, and cause problems. Can floods cause problems? I would argue yes. We have immediate deaths and injuries. We have non-specific increases in mortality. We know that when floods occur, you have massive numbers of diseases, leptospirosis, hepatitis, diarrhea, respiratory and vector-borne diseases. In some situations, you have toxic substances now in the water causing mental health effects and increased demands on health systems. Simply look at, this is from Mozambique in 2000. I don't know how many of you followed the floods of Mozambique in 2000. I'm going to guess not so many. You remember her problems in Hurricane Katrina in New Orleans? Not dissimilar, not a situation which is any different from this. So it doesn't matter if it's in a third world, developing, emerging nation, choose your term, or a first world, although friends of mine do consider the United States to be a developing country. We won't go there. But it doesn't matter. If these systems can overwhelm the ability of the healthcare system and the infrastructure to actually deal with these problems and stop transmission of parasites and pathogens. Just for the record, there are students of mine in the room. Parasites and pathogens are not the same thing, but for this talk, we will use a generic term so that I will not lose marks on the exam for using incorrect terms. 2010 earthquake in Haiti was followed by a massive cholera outbreak. Approximately 700,000 cases of cholera have occurred, roughly 8,000 deaths. Haiti had not seen or had recorded cholera for over a century. Where did it come from? Two reports. One is that these were introduced by international volunteers and peacekeepers. The other report suggests that it was a reactivated local source that had been somewhere in the soil, in the coffins, in the cemeteries, in somewhere that during the flooding conditions came out was activated. This is still up for dispute. There are groups which have sequenced the cholera found there and actually found it to be the Asian version, which was occurring in Nepal just months before the Nepalese volunteers peacekeepers came to help in Haiti. Who knows where it came from? The people of Haiti really don't care where it came from. The people of Haiti want and continue to want some source of control and freedom from dying from cholera, which is not a nice disease in the first place, under situations where we have had a massive problem, in this case followed by the earthquake. Not only in Haiti, but we know that when El Nino starts, and we know that under heavy rainfall conditions, cholera cases go up, El Nino stops, and they go down. So we know that water events, at any rate, when they increase dramatically, we have increased amount of water, we have flooding, we have contamination, we have all sorts of problems with these pathogens, which are essentially transmitted hand to mouth. You infect yourself by contaminating your hands and then putting your hands in your mouth and become infected, or drinking contaminated water or contaminated food. You ingest those. Could this happen in North America? Cryptosporidium outbreak in Wisconsin, 400,000 cases, many, many deaths in the state of Wisconsin. I don't know Okay, Arna already told you that I lived in Wisconsin. If you look on the Wisconsin license plate, it says America's Dairyland. There are more cows per capita in Wisconsin than anywhere else in the United States. This is a disease that can get into cattle, massive number of calves in the spring, massive number of parasites in those calves, got into the surface water, massive rains that year. All the water entering the treatment facilities was completely murky and cloudy. The chlorine treatment did not clear up the problem with cryptosporidium. Massive numbers of people were infected. Hundreds, if not 1,000 deaths occurred. So it's not just events which are occurring in far distant areas of the world. Certainly this can happen in North America. In fact, we had a situation in this country not so long ago, Walkerton. Now, this was not necessarily a problem only of the pathogen and water. This was also a problem of management and manipulation and us being very, very careful with what we do. So as climate change models indicate, more frequent and more severe climate events are going to occur, and our ability to deal with the consequences are going to be compromised, resulting in more disease and more deaths. And this is not just in developing countries, this is in every country. Okay, I'm going to switch from hand-to-mouth diseases, with which you sort of infect yourselves, and talk about vector-borne diseases. These are diseases, these are parasites and pathogens that are transmitted to us and to domestic animals via insects or invertebrates, sometimes ticks. The Tropical Disease Research Unit of the World Health Organization has said that these 10 are the 10 most important tropical diseases in the world. And except for leprosy and tuberculosis, the other 8 are transmitted by vectors. Nematode transmitted by mosquitoes. This is schistosomiasis transmitted by a snail. Nematode transmitted by mosquitoes, and malaria, which is also transmitted by mosquitoes. So what we're seeing as poverty increases, as climate changes increases, we're actually seeing significant increases in the problems with these vector-borne diseases, which means the vectors and the parasites are exploiting the changes in climate that we are presenting them with, and they are transmitting those parasites to us. Malaria is always going to be #1 vector-borne disease. Unless you have something else, it becomes very personal and important to you. But it kills 1 to 3 million people each year, most of whom are children in sub-Saharan Africa. All of us who write grants write these numbers in those grants to try and get funding, but these are, as far as we can tell, the correct numbers. Climate suitability is a primary determinant of whether conditions in a particular location are suitable for malaria, transmitted by a mosquito of the genus Anopheles. How many of you actually like mosquitoes? Nobody? Okay. There's one of us in the room, Peter Belton. A change in temperature may lengthen or shorten the season in which mosquitoes or parasites can survive. So what we're seeing is, with the changes in climate change, we're seeing changes in temperature. We're also seeing changes in water patterns, which means the periods during which mosquitoes survive and can transmit is being extended, and also the parasites are developing much more quickly. So we know that some changes in precipitation or temperature during the season can determine increase or decrease parasite and vector populations. In the vast majority of cases, it's increased. And it's increased because temperature goes up, which both parasite and the vector like, and increased precipitation, increased rainfall makes perfect condition, the perfect storm to increase transmission of the parasites that cause malaria. Very simplistically put, this is survival of mosquitoes as the temperature changes. As we go from 18 all the way up to 40 degrees, mosquito really doesn't care. Hotter the better, until a tipping point of around 37 degrees. So if the average temperature in an area is 28 degrees and it goes up to 30, 32, 34, the mosquito is perfectly happy. It's going to survive. It's not going to be affected. The parasite, however, is affected to our detriment, because as the temperature goes up, the parasite develops much more quickly inside that mosquito. So it used to be said that only old mosquitoes could transmit Plasmodium that causes malaria in humans, because it took a 2-week period or 3-week period for that parasite to develop inside the mosquito. Now it's down to 5 or 6 days. So what we have is that the mosquitoes are having a perfectly normal lifespan. but the parasite inside them is developing so much more quickly that we actually have more transmission events going on, simply because of climate change and weather patterns. If we look at Africa, which for whatever reason suffers more diseases than we can understand, we have the situation on the big graphs in which it is the number of months which are conducive for transmission of, for survival of the mosquito. And what we can see is on 3 different models what's going on. We see this green area extending out. The green area is the 12-month period during which transmission can take place. And in the model, it's getting bigger and bigger. The smaller graphs indicate the predicted incidence, the number of new cases of malaria caused by these changes. The darker brown means, the darkest color means we expect to see a 100% increase in the number of cases of malaria in these areas. So the mosquitoes are going to be able to live 12 months of the year rather than 6 or 7 months of the year. And we expect to see significant, I believe this is 16, 23, and 28% increases in the number of cases of malaria that we will expect to see only in Africa because of these climate change events. I'll switch to a different mosquito, one called Aedes aegypti, and perhaps some of you have heard of yellow fever, dengue fever, and chikungunya. Whereas the previous mosquito bites during the nighttime, this mosquito bites during the daytime. So we talk about malaria, sleep under bed nets, don't let the mosquitoes bite you. This mosquito bites you when you're out having fun during the daytime. And these are 3 viral diseases which this single mosquito can transmit. And with the case of dengue virus, if you look at the scenario from 1990s to 2080, if you look at South America now, we can see much more of Brazil hitting the very high transmission rates, survival of mosquitoes. The biggest concern, once again, is Africa, because we look at Africa, it's only coastal in terms of dengue transmission. Dengue is not really considered to be an African disease. It's an Asian and American disease. Under this scenario, dengue is going to go rampant in Africa. And Southeast Asia has always been a hotbed for dengue, but now the Indian subcontinent is going to be massively affected. Population of India is a billion, many of whom are completely susceptible to dengue. Jump to some of our work in South America and Colombia. So the stats will say, simple cases, that there are 50 to 100 million cases of dengue every year. 2.5 billion people are at risk of dengue. In Colombia, here in the city of Cali, where we work, it's roughly 1,000 meters elevation. Outbreaks in 2010. 2010 was a big outbreak in South America with over 1 million cases of dengue. And the 2010-2012 outbreaks, we had 200,000 cases of dengue in Colombia, over 500 deaths. But when we start understanding what's going on, we find that 30% of the vectors are resistant to dengue. They kill the dengue virus. So how do we understand the epidemiology and transmission of viral disease by this vector if we don't even know how many of the mosquitoes in a region can serve as a good vector? And I have this in red because one of my students currently in the crowd, I will not point her out, is actually spent 3 months in Colombia, is now trying to work out some of the factors that determine how mosquitoes kill the dengue virus. And should we be able to switch this from 30% to 100%, we wouldn't have to worry about transmission. And so we're trying to understand some of the molecular interactions between the dengue virus and the mosquito. That complicates understanding dengue transmission, as do the vaccine initiatives. And ultimately, we would love to have a vaccine to essentially remove people from the susceptible group and put them into the resistant group with a vaccine. Perhaps we can talk about this during the question period or afterwards, because there are a whole bunch of things going on with vaccine initiatives right now, complicated by the fact that there are 4 different serotypes of dengue virus, and if you have one serotype and get another serotype, you can actually die of what we call hemorrhagic fever or dengue shock syndrome. But Sanofi is a vaccine company from France who 2012 said they had the vaccine, then their data said, no, we don't have a vaccine. And now at the latest meeting, they said, 2016, we will be rolling out the first dengue vaccine. So maybe 5 years from now, we won't be talking about dengue whatsoever. We might be talking about chikungunya. How many people have heard of chikungunya? How many people can pronounce chikungunya? And tell me if I'm pronouncing it correctly. I don't know. It's a viral disease, again, transmitted by the same mosquito that transmits dengue, found in Tanzania in the 50s. As of I'm going to say, for those of you who've heard about chikungunya, how many of you knew about it a year ago? Not that many. It entered the Caribbean. 34 countries now have confirmed cases. There are 1.2 million suspected cases, 24,000 confirmed chikungunya cases. The reason that we don't have as many confirmed as suspected is because it's come in such numbers so quickly, it has completely exhausted the ability of healthcare centers to determine what you have. You're walking in with a viral disease with these symptoms, and they say, you have chikungunya, take this drug. Great. We actually don't have a drug to kill the virus. We have drugs to make your symptoms better. The trouble is, if in fact it's not chikungunya, but dengue, which is very difficult to determine which it is, unless you do proper lab tests, some of the drugs you're getting for chikungunya can drop your blood pressure, and if you have dengue and you're moving towards dengue hemorrhagic fever, you don't want to drop your blood pressure. So if you have dengue and they say you chikungunya, and they take the drug they tell you, it can be bad news. So understanding what's going on is essential to our understanding of how to treat, but at the same time, we actually don't know how to deal with massive epidemics of a couple of months, well, less than a year, over 1 million people walking into clinics and saying, I've got the disease, what do I do? What would happen if we walked into your healthcare provider, even this many people in the room, and said, right now, let's go to the emergency room, all of us, and say we have a viral disease, and what would the health people be able to do? Take that now to an emerging, developing nation where they are struggling as it is with a very limited healthcare budget of $100 to $500 per person per year, and all of a sudden you get thousands of people walking into a clinic. How did chikungunya get to the Americas? The simple answer is we don't know. The sequencing of the serotype of chikungunya, which appeared in the Caribbean, is very similar. Essentially, it is a Southeast Asian version of chikungunya. It appeared in a small island in the Caribbean. The first report was in the island of St. Martin in the Caribbean, which is a joint French and Dutch island. And it's believed that a French tourist went from Southeast Asia or French Polynesia back to France and then now St. Martin. And from that one event, which we believe is true, we don't know, of course, the 1.2 million cases of chikungunya virus which have been suspected in the last 8 months. First in the Caribbean, then into Central America, and now that we have hit our winter, which is the summer in the southern hemisphere, it's massively just running through Brazil, running through the Guyanas, Venezuela, Colombia, it's everywhere. What do? We spray. How many of you would be adverse to killing mosquitoes? How many of you would be averse to killing all the beneficial insects by using insecticides which have no specialized effect upon mosquitoes and are killing all the beneficial organisms? Some hands go up. So now the question is, where do you weigh the balance in a limited budget area where 1.2 million people are suffering from a disease? We have insecticides, we have consequences. So every time we have an event, the movement of parasites or pathogens to a new area, we have the weather conditions which allow those to develop and flourish and infect us, and we have abilities to try and reduce those, rather ineffective in many cases. Where do we invest our money to try and mitigate and try and soften the effects of these factors? When we start talking about vector-borne diseases and the effect on climate change, the problem that some of the politicians and decision makers are saying to us is, we want facts. Don't give us ranges. Don't give us probabilities. We want facts. If we do this, what return will we get on our investment? And when we can't make predictions exactly what the temperature range change is going to be. We have to put this into our model and have 95% confidence intervals to try and figure out what is really going to happen. So we have uncertainty in our projections. We have a nonlinear response. Temperature alone is insufficient to make accurate predictions, and we know that from our models based upon one of these temperatures up here. And then we have evolution. The pathogens and the vectors are adapting physiologically and genetically, not only to survive under climatic change and differences, but also to flourish under those conditions. So things are not static either in the climate or the biology of the organism. This slide is simply to show that vector-borne diseases are massively important, probably responsible for the deaths of 5 to 6 million people per year, and climate change and variability has a major effect upon them. But don't forget, there are a whole bunch of other biological, abiotic factors and sociological factors that we have to deal with if we're going to try and reduce vector-borne diseases. So when we go to these vectors, if you increase the temperature, you have faster development time of the parasite. That means transmission goes up. Warmer temperatures can shorten the vector development time. Transmission goes up. Increased temperature usually decreases vector survival. So vectors, when it gets too hot, they don't survive well unless it's really humid. If it's really humid, they don't mind the temperature. All the predictions of the models will tell us not only is temperature going up, but humidity is going up as well. So it depends on the geographical range of the vector, host, and parasite. Most of the models say transmission is going to go up. So when we get asked, I get asked, what's going to happen? I say, you tell me the exact conditions in the future, the vector you have, the parasite is transmitting, the geographical region of the world. I'll tell you with great certainty what's going to happen with this vector-borne disease. But making global predictions on a long term is really difficult and it has been hurt quite badly by certain models not being accurate, which means some people say, well, if that model's not accurate, which model is? And so we suffer from the fact that, unlike the weatherman, people expect scientists to be right all the time. I've touched on this subject a little bit before, migration and movement of people and disease transmission. And this is quite a nice little book to read on migration and health. We have natural disasters. We have terrible conditions. We can have drought. We can have floods. We can have whatever condition. People move. People move to better places, whatever a better place may be defined as. They take the parasites and vectors with them, not on purpose. We're not talking about taking your pet dog with you along the trip. We're talking about by accident. And then when we get to these new locations, we have transmission to people. Sometimes people who had never seen these parasites before, they have no immune defenses, they are completely susceptible. And so this is not done on purpose, this is done by mistake. And when we look at migration of peoples, we know from gene studies and looking at insects along this transect alone from Colombia and Brazil and Venezuela, we find Colombian, Venezuelan, and Brazilian insects up here in Mexico, up in the Yucatan. And there is a pathway of migration up here through which people are migrating or emigrating or fleeing areas of civil strife or whatever and moving to the United States to improve their lots in life, and they're taking with them all sorts of vectors. We can follow those trails, and we know where they are based upon where the species of parasite and the species of vectors that we're finding along the way. In the United States alone, over 100,000 people are estimated to have Chagas disease. Chagas disease is a disease found only in Latin America. And the vast majority of people who have moved up from Latin America emigrated to the States, some legally, some illegally. Estimated 100,000 cases. There are estimated 10 to 15,000 people in Canada who are infected with Chagas disease, often found out during blood donations, otherwise not tested for. We actually don't know how many people have it because there is a stage of this disease in which the parasite disappears. You can't find it in the blood. And then after a period of inactivity, you come down with heart trouble and die from a massive heart attack caused by this parasite. So it's a silent disease, silent until you come down with a heart condition. Let's switch to something really much better news, and that's Ebola. How many of you have heard of Ebola before a year and a half ago, 2 years ago? Okay, there. I now know how many people have taken my course. It first appeared in the 70s. It's called African hemorrhagic fever, acute, mostly fatal. high fatality rate. From the 1970s until a year and a half ago, there were roughly only 2,500 cases reported and about 1,600 deaths. From the 1970s until about 2010 or whatever the last outbreak was. Current outbreak, which is not yet ended, we have over 22,000 cases and over 9,000 deaths. What's happening? How is transmission taking place? What's going on? Well, we know some of the things that are going on. We have people who are, this is usually a disease of very small, very remote communities. People will go and become infected, and we'll talk about how they become infected in a moment. They come back to the village and they have the symptoms, and anecdotally I've been told by some people that in these very small communities, if you come down with these symptoms, the community says stay in your home. If you survive, you survive. If you die, you die. But you're not going to come out and infect everyone else. The community will bring you food in some certain situations. This Ebola outbreak got into the urbanized situation and got person-to-person contact, massive transmission. It's still going on. And I'm not sure if this were to arrive in North America, how we would deal with such a massive number of cases of Ebola. But from a biological point of view, there definitely is a clear link between human infection with Ebola and human proximity to primates. Outbreaks have occurred in countries that house 80% of the world's remaining wild gorilla and chimpanzee populations, and Ebola is terrible on gorilla populations. So some of the reports of how humans become infected with Ebola, the first cases, people are hunting in forests, people are extending themselves into new areas, coming in contact with wild animals such as gorillas, eating bushmeat, becoming infected, and going back to their community and taking the virus with them. And the situation is such that humans, and this disease itself is not directly, probably, affected by climate change, but it is indirectly in how other animals, such as the gorillas and the other primates, are affected by climate change and their migration patterns and their events in which there is contact between humans and these primates. The biggest question we have is, where does Ebola hide in non-epidemic years? It disappears for regions for 10, 15 years, and then reappears in a burst of activity and disappears. We have no idea where it hides. A number of years ago, the literature was full of news that it's fruit bats. Fruit bats cause Ebola. Fruit bats transmit Ebola. Yes, bats will have antibodies against Ebola. Any human who survives also will have antibodies against Ebola. We can find Ebola DNA sequences inside the bats. As far as we know, they don't show any symptoms. It's considered the best candidate to be the reservoir. But really, we're guessing. We don't really know the contribution of fruit bats. And when you look at the variant of this Ebola virus, if the fruit bats were to have flown from the location where they were supposed to be, based upon the sequence of the Ebola, well over 1,000 kilometers over to the part of West Africa where we have the outbreak right now, the situation is how come along that transect there were no cases of Ebola moving along? So we don't really understand Ebola. We don't understand understanding climate change. We don't really know how climate change will directly affect Ebola, but we know it affects some of the other primates which are implicated in the transmission from the wild, from the feral situation into the human population. And if you want definitive answers on all the data, you can go back to the 1990s and read these really great books on everything you want to know about Ebola. But hey, you can watch the movie if you want to. So, new diseases. This is what the scientific community is quite worried about. We cause disruptions in ecosystems. Pathogens jump from animals to humans, and humans spread these widely. So we go into situations where we upset the ecosystem, we have logging, we have slash and burn, we have agricultural activities, we hunt and eat wild animals, perhaps picking up Ebola, we damage the ecosystem, and we say it's a density-dependent relationship because 7 billion people doing this have far more effects than 7 million people. And within these new areas in which the people are going and carrying out this activity are viruses, bacteria, fungi, which emerge. They emerge from where we had never seen them before. Millions of these organisms are parasitic. Organisms never before been seen or identified by us. So all of a sudden now we're coming into contact with brand new organisms. Trees are demolished and cut down. What was 50 meters high in the canopy is now at ground level. And it's believed strongly that this is how yellow fever, a virus spread by mosquitoes, got into the human population from primates that lived on the top of the canopy. Was we knocked down the forest, there were no more trees with high altitude for the vectors or the primates, so the vectors actually bit us and started yellow fever outbreaks. We can eat the meat. Pathogens, the parasites or vectors or whatever is there is deprived of the preferred host, and they jump onto us. So they're not targeting us. They're not saying, I'm waiting for those people to show up, but they're certainly exploiting our availability and our abundance. Okay? And this is, without a doubt, the emergence of new diseases, new if you want to call them that, are a direct result of our actions. I mentioned Chagas disease earlier, which causes the heart problems. This was a disease of wild animals in the forest. We never knew about it. We never saw it until we started chopping down the forest in Latin America. Actually, we know it. It's been around for an awful long time. But as soon as we created human habitations and scared away all the other wild animals upon which the insect fed and the parasite replicated, and brought in our own animals that live around our homes and in our homes, it became a human disease. And there are certain vector species and parasites that now never leave the home. They used to go out, fly back to the forest, and come back in, and now they are 100% adapted to humans living in houses. And if you look at some of the work detailed over the last 65 years, again, a statistic I did not just make up, 71% of the new diseases come from wildlife. So we know historically, through time, many of the diseases, parasites, and pathogens that we harbor that cause us disease, we've obtained from wildlife situations. Many of which are coming from Southeast Asia, up in the forested areas. Okay? And the argument is that if we can have conservation efforts to reduce the contact events between people and these wild situations, we probably could limit, or reduce, not eliminate, future outbreaks. But we know that avian flu, SARS, human flu, swine flu, MERS, Chikungunya, Ebola has originated elsewhere in other hosts and jumped over to us. And we are putting pressure upon the system to turn around and try and clear these problems. Climate, infectious disease. Climatic conditions set the geographic and the seasonal limits of potential transmission, whereas other factors, social, behavioral, public health strategy, determine where and when real transmission takes place. I'm not going to walk you through this, except to say that climate change impacts on the physical systems directly, on biological cycles directly, or indirectly on human health, which can be exacerbated when they already have other diseases or malnourishment or other disorders, and they have a major economic and social impact, which is ecologically mediated or socially mediated. And it's easy for me to say, I'm a scientist in my specific area, and I don't want to understand those other areas, because I want to be the king of my little fiefdom. But what we're finding out is that these, in fact, are integrated, interdisciplinary effects, which need to be studied by multidisciplinary teams. This is not how we have to speak to politicians. And I'm going to guess that half of you said, what the heck's he talking about? And I don't understand. Arna understands these things. I don't. But this is not how we should be talking to the decision makers. We point out the trends, the possibilities, probabilities, hypotheses, and we tend to use a lot of gobbledygook. But the politicians and decision makers want 100% facts. They want us to say, this is happening, no error bars. So our job is to educate the politicians. Their job is to listen, to learn, and respond, and to take action. And we have to give them, we have to learn to speak their language, and we have to give them the facts, and we have to persuade them that, in fact, we have to deal with some of these problems, or else they're going to get worse. We know that climate change is occurring. We know that climate change has massive effects upon disease transmission, but there are other factors as well. Population density, urbanization, especially in some areas of the world, massive density, massive urbanization, people fleeing from civil strife or droughts or floods. We have all sorts of other factors involved. Climate change is a major one, but not the only one. Some conclusions I'll leave you with. Climate change is real. Don't doubt it. Don't listen to the naysayers. or listen to the naysayers and provide them with alternative explanations. Short-term predictions are difficult to make, and we're going to be wrong on our short-term predictions, such as the weatherman is often wrong in what's going to happen. But long-term, we're pretty sure they are real. We're going to see increasing severe weather events, which become more and more common. People are going to move to safer areas, and they're going to move with them sick people, pathogens, parasites, vectors, coming into naive hosts. And this is not only moving from a country with problems to Canada. This can also be within a single country where these diseases are limited by the presence of the vector. So in parts of Costa Rica, excuse me, Michael, parts of Costa Rica, we know the elevation limit of dengue transmission, which is defined by the altitude limit of the mosquito. The mosquito won't live more than, let's say, 1,000 meters in altitude. What's happening now with climate change, global warming, the temperature ranges are moving up the mountain. So now we have more and more parts of the mountain are suitable for transmission. People who lived at 1,500 meters may now be bitten by mosquitoes which are residing there, causing transmission of dengue. So we're seeing not only a north to south increase in disease transmission, we're also seeing within the same country at an altitude difference. New parasites and pathogens are being transmitted everywhere. Some condition will increase vector and parasite populations. We know that. Crowding, malnutrition, lack of drugs will affect specific populations, but population stressors are going to force people and pathogens into contact. And what we have to work on is mechanism by which we can eliminate that. We are going to see new diseases. Monitoring and surveillance are essential, but they're often neglected for various reasons, many of which are financial. And we have to learn to speak the language of the politicians to educate them and persuade them to help us take action based on evidence. Evidence-based action. There are certain political groups who don't like to listen to evidence, would rather, well, we won't go there. It's not a political discussion. But we no longer believe that diseases are caused by the gods being angry and venting their wrath upon us. We actually do understand what causes disease and the effects of real and predicted climate change on this transmission of these diseases. We understand, in many cases, how to reduce disease. We now have to act upon this knowledge, and if we don't, all we're going to see is massive increase in illnesses. Understanding climate change in disease interactions is formidable, mainly because of the interdisciplinary nature of the interactions among hosts, parasites, and ecosystems. Many of these interactions can covary with climate change scenarios. Effective forecasting of climate change impacts on disease requires filling gaps in data, theory, and scale, and the effects of hand-to-mouth diseases are going to be very different from vector-borne diseases, as examples. And all our claims that we say are going to happen have to be tempered and supported by scientific rigor, real data, open-mindedness, and broad thinking regarding these crucial issues. And the problem sometimes we face in the scientific world is that I can work on a very small area, a very small project, get my grant, get my publications within a time period to get another grant. And that's how academia tends to work. These very complex interdisciplinary and multidisciplinary studies take a long time to set up, a long time to carry out, a long time to analyze, and if we continue with the current form of rewarding professors by publications, and it takes much longer to do this, this work may be more important, but we don't get rewarded for it. Sometimes all the pieces of the puzzle seem to fit together and tell a story when you start to work together. Sometimes not. Sometimes you stare into empty space and say, we were so close. We're just this far away from really understanding what's going on. And what we really have to persuade people in the multidisciplinary areas is to help us fill in the void, to help us understand the complete situation. I don't work alone. I have to acknowledge a whole bunch of extremely bright students, undergraduate and graduate I've had at SFU. Very, very strong collaborations, mainly in Latin America, where we work a lot in Colombia and Brazil, and by very wide and different funding agencies, Canada Research Chairs, NSERC, Colciencias, which is the NSERC equivalent in the country of Colombia, Bill and Melinda Gates Foundation, CIHR, and the Michael Smith Foundation. So I'll leave you with that. I don't, as much as I've given you gloomy prediction, there are a lot of solutions out there. And so it's our job to persuade people to let us work with the solutions, find the solutions, and carry them out. Thank you.
Everyone can take a breath and stretch. Think a little bit. All right, that was a lot of data, a lot of ideas, and I expect there are going to be some questions. So put up your hand, and Laurie will give you the mic. Who has the first question? I've got a question, but I'm going to wait.
There are students, past and present, from my classes who should have lots of questions.
They have
or
answers.
people hiding in the back row like Sebastian.
Or Sebastian. People still gathering their thoughts. I have a specific question for you, Carl. Aedes aegypti is a mosquito that carries the not the malaria, but the like dengue fever.
Yes.
A lot is known about the biology. When is it going to arrive in Vancouver?
As some people in this audience know, Aedes aegypti is already in Vancouver. There are about 300,000 individuals in my lab at the moment, in my insectary. If there is an outbreak of dengue or chikungunya in Vancouver, you know who the press is going to call. They're going to call me. But realistically, I believe the conditions are not good in Vancouver for Aedes aegypti. There is another mosquito called Aedes albopictus, which can also transmit both chikungunya and dengue, and it probably could survive quite well in the Vancouver conditions. Now, it also is one of the major pest species reported by the mosquito control groups in the East Coast of the United States, from New York all the way down the coast. It's #1 nuisance mosquito. So the big fear in the Eastern United States is that chikungunya this summer is going to hit the lower 50 states, and Carolinas up to New York. It's going to be like West Nile virus was back in the year 2000, 2002. But we, for whatever reason, probably are going to be spared.
But mainland America is not going to be spared.
The East Coast will probably have massive chikungunya within a year.
Joan.
I have a question, Carl. I'm curious. I don't know if this is true, but what I was told, what somebody told me was that there was quite a fear that West Nile would come here, and because that led to, it was the West Nile that led to the crow deaths, right? And I know in my brother's neighborhood in Toronto, they lost 95% of their crows. And I was told that the reason it didn't make it out here was that we simply don't have enough mosquitoes, that there was not enough mosquitoes to actually support the pathogen. Is that correct? And if that mosquito, if West Nile wasn't able to make it here because of mosquitoes, does that mean that we're sort of safe from all kinds of mosquito-borne diseases?
Right. So the question is, why didn't West Nile make it here? Was it because there are not enough mosquitoes? And if that's the case, are we safe from everything?
You heard it here first, you're safe. Michael, how many mosquitoes have you sampled over the last 10 years? Millions. We have mosquitoes here which are perfectly great vectors of West Nile virus, of the genus Culex. Why we don't have West Nile is something a lot of people are working on, including a student of mine, a mathematical modeler. There are several reasons we think. We know that it came west and probably hit the Rocky Mountains and never went over. However, it did go south across to Texas, California, and came up. Every year there are lots of cases in California, fewer cases in Oregon, a couple of cases in Washington, and about 4 or 5 years ago there were 2 cases in the Okanagan. But that's as far as we know, 2 cases. Crows die from this massively, but if you talk to people on the East Coast of the United States where it first entered, the crow population crashed. And now it's built up again by individuals which are resistant. So it's what Toronto is also going to see, much to the chagrin of the people from Toronto, okay, is that their crow populations are going to recover. we have a different species of crow here. So I don't know if the species of crow here is as susceptible to dying as are the eastern crows. And this virus requires a lot of bird infection to amplify, to bump up the number of virus particles in their blood to infect the mosquitoes. So if we don't have hosts which can really amplify to large numbers, we'll never see a major outbreak. But there's no reason whatsoever why West Nile virus should not be transmitted. We have the temperature regimes, we have the mosquito species, we have everything which should create conditions for transmission, but we don't see it. We're the only political jurisdiction in North America, except for Alaska, which doesn't have it. Or it could just be because we're so laid back and relaxed in BC that, you know, it's not worth the virus's trouble.
Yes, sir, Michael.
Carl, I just want to ask you a question just to sort of clarify some things which the audience might be thinking about. In terms of not all mosquitoes, there are all these different species of mosquitoes, but not all of them carry the same diseases, and quite often it's very specific to one species, and there's like 100 species. There's 50 species here in BC or more. More, because we found another one.
Here.
Yeah. So why is it that some species of mosquitoes carry some diseases and others don't?
Thank you for asking, Michael. It's almost as if I planned that question, which I didn't. The interaction between any pathogen, can be malaria parasite, can be dengue virus, has to do with how it interacts on a molecular basis with its host. And so many insects, an insect can bite a person who has 25 different parasites in them, but a specific mosquito will only transmit one of those parasites. Perhaps it's highly attuned and evolved to transmit that one. It will kill all the others with components of its immune system. So either it turns on its immune system to kill it, or the pathogen, the virus needs a special receptor, a special doorway to get into the tissue it wants to go into, and that receptor is not there. So we know without a doubt that only certain types of mosquitoes or kissing bugs or whatever will transmit specific parasites. And we can actually determine or predict what's going to happen based upon the presence of suitable or non-suitable vectors.
I'm just wondering if you could talk to us a little bit about what are the best places for intervention in terms of dealing with this problem. You know, if you had to pick three, for example, like you've talked about the web of causation and the multiplicity of factors, but if you were speaking to a government official today, and we wanted to start to solve some of these problems in the next couple of years, where should we invest in terms of dealing with this?
So if someone just phones you up and says, you're on the panel, we got vector-borne diseases here, what should we do?
I think what you should do is give me a massive research grant.
Scratch that from minutes.
Tell me which vector or disease you're talking about, and then the response is going to be different depending on what we're going to do. If it's a waterborne, it's different than if it's a vector-borne.
Okay, let's say vector-borne, for example.
Dengue virus. We don't have it in Vancouver, we don't have to worry about it. But if you are living in Colombia, you want a Canadian context, or do you want a...
Here. The federal government just, yeah.
West Nile virus. Yeah. It's in Canada, what to do. Monitoring is essential. We have to know if the virus is present, if infected birds are present. We have to have massive monitoring programs. We have to know what's going out there. We can't deal with any efforts whatsoever until we can turn around and we can say, this is the real situation. Not a predicted situation, a real situation. Subsequently, massive efforts should be going into vaccine development, because if we can vaccinate people, vaccines are not the overall solution. There's still a whole bunch of problems associated with vaccines, what's going on right now with measles outbreaks and all the rest of it. But if we can understand the biology and develop a vaccine, we can essentially eliminate, for all intents and purposes, the problem. So understanding the basic biology of what's going on, finding the weak points in that life cycle, and then attacking it. And with viruses like West Nile virus, probably the vaccine is going to be the solution.
It's a technical solution. Up here at the back.
I'm curious to hear your thoughts on them releasing genetically modified mosquitoes to control disease-carrying mosquitoes.
Did I say that?
No, but we'd like to hear your thoughts on releasing genetically modified mosquitoes to control mosquitoes, right? This is to bring the mosquito populations down, right?
Yeah.
Yeah.
Okay, there are 3 or 4 different models which are being used right now to genetically modify insects to reduce transmission. One is called the RIDL method, in which mosquitoes which transmit, for instance, dengue, are raised in a situation in which they have antibiotic in their diet. They have a lethal gene that as long as they have the antibiotic, they don't die. They're released as adults, they mate with the wild mosquitoes, all the offspring don't find antibiotic in the aquatic environment, all of them die. So if you release 100 million mosquitoes like this every week for a number of weeks, the population of the wild mosquito is going to crash, but it's a massive investment and a constant introduction. The other model, another of the models, is transgenic mosquitoes which produce compounds which kill the parasite they're trying to transmit. And so a group from California, Tony James, has a mosquito, Anopheles stevensii, which is a vector of plasmodium, which causes malaria in Southeast Asia, which does not support the development of the parasite. So what they're working right now on the logistics and government permissions to release a transgenic mosquito, which has a whole bunch of social problems, and people don't want transgenic organisms. The third, and perhaps the best for the moment, is a group out of Australia who has Wolbachia, which is a bacterium, put into the genome of the mosquito. And it took, as the researcher who did this told me, he burned out 13 postdocs, and I don't know how many grad students, trying to get this to work, because he said, it's got to work. And eventually it did work. And the Wolbachia bacterium is transmitted from female to all her offspring, and it has also something called cytoplasmic compatibility. So if a male mates with a female, and the female has Wolbachia, all the offspring will have Wolbachia. Doesn't matter if the male is infected or not. But if it's the other way around, if the male's infected and the female's not infected, no viable offspring will result. What it means is that this is a driving force to get Wolbachia throughout the populations. And this has now been released in Australia. Studies are undergoing in Thailand and in Kampuchea and now in Brazil. And the whole concept is this Wolbachia actually shortens the lifespan of the mosquito and reduces vector development. So mosquitoes live a shorter time, virus doesn't develop, and so the probability of transmission is significantly reduced. All of these have social implications because people don't like the concept of transgenic organisms, especially if you go to Europe or certain parts of the world. They don't want transgenic plants, they don't want transgenic insects, and so it is a logistic problem, as Dr. Johnson was mentioning a moment ago. How will we deal with this? This is one of the most promising projects to eliminate vector-borne diseases using a bacterial symbiont, a bacteria which lives inside the insect. Okay? And this was funded, just saying, by the Bill and Melinda Gates Foundation with a $10 million grant to this group in Australia who said, we can do it. And the Gates Foundation takes chances on risky opportunities, which probably won't work, but maybe they will. And this one, it looks like it actually did work.
So can I just add another comment here? I think it'd be interesting, I don't know the answer, but it'd be interesting to ask people whether they care more about transgenic food that they eat versus transgenic mosquitoes. Maybe the barrier is lower, but I don't know that. Okay. Next question or comment from the audience? Yes.
Could filth flies, such as house flies, be contributing to the spread of Ebola?
You're on camera, just so you know.
I know. Yes. A group of us got together and looked at what's happening, and absolutely we believe that the house flies and filth flies could contribute to it, contribute to transmission of Ebola. There was a group at the Entomological Society of America in Portland, Oregon, which was just last November, which actually had a working session on this. And the conclusion from them was, no, it's probably not going to happen. But there's no reason why it couldn't. Because if you can transmit all the bacterial and diseases or pathogens which occur in manure, in dirty conditions, you know, all of us, we go to that church picnic or whatever out at Bunsen Lake, and you got the flies on over where the dogs are doing whatever dogs do, and then they come and land on your potato salad and they give you insert salmonella, Campylobacter, pick your disease of choice. There's no reason why in humid Africa, in situations where people are bleeding out, they have liquid from all the orifices, it's virus laden, there's no reason why a fly couldn't land there and land on someone else and transmit it. So I suspect it has happened, but at a low number of events and probably is not the major route of transmission, but I have no doubt whatsoever, yes, it could happen.
So just as a follow-up, so why did the working group decide it couldn't, if you're so adamant that it could?
Because they're working on probabilities. They say, oh, the virus won't replicate and won't do this. But you're talking about a closed house, a closed hospital room, open windows, flies going in and out. In a hospital setting where we have sterile conditions, much less likely. But in a rural area where there is no control of this, perfectly possible.
Good question. Another other question. Yes. Shall I run around, or is Laurie going to go? Laurie's going to go.
Hi, thanks for your talk. This is sort of following up on the question about where do we focus, and more so along the lines of novel pathogens. So we're at this really exciting time now where we can use next generation sequencing and all these high throughput techniques to go out and look for new pathogens that might be a risk for human health. And I'm curious as to where you think we might focus best, because that's a lot of money and it's also a lot of data, and that's a real pain to sift through. So would you recommend looking at vectors, or maybe animals that are in close association with people, or something along those lines?
Right, so where do you look for new diseases?
Next-gen sequences is a tremendous tool, and one of my students in the audience is now trying to work through 250 million different sequences from our mosquitoes in Colombia. It's a computational situation. It's no longer a biological question. If you want to go out into the mountains of Nepal and start looking around for potentially new pathogens you might encounter in those mountains, yeah, I think you're really hoping that you're going to find something directly. The areas to look, in my opinion, would be in the margins of where people are going into remote areas, those areas of the borders between human settlement and completely feral conditions where we don't normally have people going in, and watching for activities where people are undergoing logging and are essentially exploiting and changing the environment in those very marginal areas. As you know from next-gen sequencing, you will generate so much data, you may spend years trying to analyze it. And you're going to get results, you're going to get data, you're going to get sequences. Now, when you align them with RNA's phylogenetic abilities, you're going to say, oh, this is the closest to Clostridium we've ever seen, or it's as closest to West Nile virus we've ever seen, but in fact, it may be completely irrelevant. So I would say that monitoring people and domestic animals in the areas around those borders between what we have massive urbanization and the complete feral condition will tell us what are actually getting into humans and actually what might be causing disease. But again, it's a massive investment. When Joy asked, what would we do? We can't put soldiers with RNA-seq machines or next-gen sequencing machines all up and down the Thai border saying, we know something is coming here, we just want to catch it on time. We have to be able to, once it does get there, use next-gen sequencing, whip through it as fast as possible, find out what it is, and then look for strategies to reduce it.
So early detection. Not prospecting. Another? Yes, down here. I got it.
So it seems like transmission rates are expected to go up for a lot of these diseases. Is there any evidence that they are perhaps evolving faster as a result of higher transmission? And if so, do you think any of them would evolve past current treatments or current vaccines?
What are you talking about? The pathogen itself?
Sure.
Sure.
So, current evolution, how does climate change affect the evolution of these parasites?
We are altering the evolution of parasites simply by putting a major stress upon them by our drug treatments. Right? So we are inducing adaptation and evolution of enzymatic systems, of sodium pumps to pump the drugs out of cells and all the rest of it. So we already know that Plasmodium causes malaria has a tremendously good system which has evolved to get rid of the drugs we use to kill it. So are they going to, evolve themselves out of existence? No, they're extremely content where they are. They're extremely successful. The pressure we put upon them are in fact causing modifications both in the vector and in the parasite. We can't predict how. We can predict if we give them a drug treatment that with time they are going to develop with a short time, they will develop the mechanism to either detoxify the drug or eliminate the drug. Okay? But I don't think we're going to actually cause a major difference in transmission rates by, I haven't changed my mind. I'll go either way. It depends on the parasite you're looking at, right? And I'll say that. The viruses we talk about are constantly mutating, and we know that at massive rates compared to the Plasmodium. Plasmodium is actually pretty stable.
Right. So the fact that, the question at the back, way at the back, yeah, Sebastian.
Thanks. Hi, Carl. I was thinking about bed bugs and the paper published last year on the capacity of bed bugs to transmit Chagas. Do you think bed bugs would benefit a lot from climate change since they're mostly domestic? And I don't know how much of Chagas prevalence in the States and Canada is explained by bed bugs or just it's brought through the States mainly because we just found out that they could potentially transmit. I don't know if they actually do, we just know that they could. So my question is, do you think bed bugs will
do better with climate change?
Yeah, and maybe some of the unexplained cases of Chagas in the States and Canada could be explained by their capacity of transmitting this disease.
You could expand that question to how does climate change, how do you think climate change might affect those diseases that are domestic, like Chagas?
I'll start with bed bugs. For those of you who haven't seen the publications recently by Gerhard Gries in the news, and now we have these compounds which can, essentially attractive compounds of bed bugs for trapping mechanisms. Bed bugs are, as you well know, Sebastian, are highly attuned to human development and human activities. And so essentially what they are doing is they are exploiting human activities and the conditions we provide for them. Probably irrelevant of climate change per se. Okay? Because they live so closely with us, as long as we're alive and living, bed bugs will do very well. Okay? Now, in terms of transmission of something like Chagas, the paper that came out was the first paper ever, I believe, to suggest that bed bugs could transmit Chagas. And bed bugs are it's interesting because all the other studies which have ever been produced suggest that bed bugs do not transmit pathogens or parasites to humans. And it's been one of those things which has been studied, and we have no idea why not, because they should. They feed on blood, they bite one host, they go on pick up the parasites, they could bite another host. There's no reason why they should not, but they don't. So we believe, as Michael asked earlier, what's going on, we believe that immune factors inside those bed bugs probably kill the parasites. Now, the study you talk about suggested that they could transmit Chagas. I think I'd like to know if that is a specialized lab colony of bed bugs or it's feral. The vast majority of cases of transmission of Chagas in North America are confirmed by people who've been to Latin America, who have emigrated from Latin America. And if it were a massive case of bed bugs transmitting Chagas, there are 25 to 30,000 people infected with Chagas in Spain, Latin immigrants going over there, and there's, as far as we know, there's never been a natural transmission of Chagas in Spain by any form, except by blood transfusion, and definitely not through bed bugs, and there are a lot of bed bugs in Spain. So going back to this, climate change will affect the vectors which live in houses with us by the sense of affecting how we perceive, how we do things, but they are going to be, I think, buffered quite a bit from environmental climatic differences because we're trying to maintain our house in a situation of stability, and they won't be subject as much to the great variations in ecological parameters.
Are there projections for climate change projections that suggest that Chagas will start to be transmitted in North America?
Oh, it is. It already is.
You said that all the cases come from people coming up from Latin America.
Vast in Canada.
Yeah.
In the States, there are vectors living. There are vectors which have probably moved up. This one right here, Rhonius prolixus, is probably moved up from Central America. It's found in Texas. It's found in Florida. It's found in Louisiana.
Okay.
Many of the dogs in certain areas are actually seropositive. So we know it's moving north. There are reservoir hosts of Chagas disease, which are essentially animals in which the parasite can develop and do perfectly well. How many of you have ever seen an opossum in Vancouver? Nobody? Tremendously great reservoir host for Chagas disease. So we have potential reservoir hosts. We don't know if the ones here are infected. I don't think anyone's studied it. We have the vectors moving up. We actually don't. We have similar insects, but not good vectors of Chagas, so we probably are okay here. But without a doubt, they are moving up in the United States.
We're okay here now, but
No.
okay. Sorry.
Housing is a big issue these days, it seems, in many regards, and especially with the idea of densification, which you touched on a bit in these terms. About, do you have any idea of what kind of densified housing would be, I don't know, bug resistant?
Sorry. The question is, how will increased urban densification affect disease transmission?
Yeah, or what would be a good model as a condo development better than single detached housing in terms of transmission and...
Oh, right. So if you're an urban planner and you're worried about disease, what would you do?
Once again, going back to the question, it depends on the pathogen, it depends on the disease. If you have a perfectly clean water supply, even with high density, you're probably not going to have the waterborne diseases causing problems. If the water supply is contaminated, it doesn't matter if you live in a detached home or in a building with 1 million people. If the water is contaminated, you will have infection. We do know that in many diseases, it's a function of density, a function of people crawling over each other to get to places of contact with people. So in those diseases, absolutely, high density will result in higher transmission if a parasite or pathogen is present, without a doubt.
And that was effectively the Ebola situation in West Africa because it got into the urban centers.
Which is also the dengue situation because we've got, dengue is not a wild forest disease, it's an urbanized disease. It's now completely urbanized by the mosquito.
We have a question here. I just want to make sure there's no one. Yes.
Carl, I have another. I can't let you get away without talking about politics in all this, because very often, I mean, we're on the sort of we're on the front line with all this, and it's like it's always reactive. That's what it is, and so something happens, and everybody suddenly goes crazy, and no one knows what to do. And the Ebola situation is a perfect example of that. West Nile was the same here. And nobody wants to do monitoring. Politicians hate monitoring. Accountants hate monitoring because it is an ongoing cost. But it's a hell of a lot cheaper than having to spend some vast amount of money running around like, you know, blue-ass flies, basically, and spending a fortune. So how can we get scientists to really promote monitoring and keeping an eye on things? And you can monitor these vectors. We knew there were bed bugs. We knew there were mosquitoes that carried these diseases. And yet everybody has to race around at the last minute and try to do something about it. So how can you make monitoring like acceptable to politicians?
Your first question asked, what about politics? And there was a picture of Rick Mercer up there a little bit earlier. And if you listen to one of his rants, for those of you who watch Rick Mercer's rants, he said politics. Poly, very many. Ticks, blood-sucking parasites. So it's a bunch of blood-sucking parasites, politics. However, they are a necessary evil in this society or a necessary great situation to deal with. We know from experience that monitoring is the key. It doesn't matter what you have to know what's there, how much of it's there, when it's not there. The problem with monitoring is it's a massively expensive program in non-epidemic years. So you could monitor Ebola in Liberia for the last 20 years and you would have had all that expense and you would have never seen a thing. And then one year it flares up and people say, what the heck were you doing? You're supposed to be monitoring that thing, right? And so it's extremely expensive to do. It's necessary. Now, we have the liberty in richer society, richer countries, to say we're going to invest in that. We're going to invest in monitoring procedures to make sure parasites and pathogens don't come across the border from Mexico with bacterial diseases on them. And we can have CFIA measuring every 1,000th strawberry to see what's going on. In many parts of the world, there is not the financial ability to monitor diseases which may or may not show up every 5 to 10 years. It's essential. It's great to do, great to have those data, but in many parts, it's just so difficult to persuade people to invest in something for which there probably is no positive return.
So it's difficult. Okay, we'll take 2 more if we don't. Yeah.
Yeah, thanks. So just to follow up on that, I'm curious how effective monitoring is in developing nations. I'm sure it's probably a bit more effective in somewhere like here, but the issues with Ebola in West Africa were not just climate-based. They were also due to socioeconomic and political factors. You can probably monitor all you want in Liberia, but if you see it happening and you can't stop it, what good is the monitoring in the first place?
Absolutely. So as I said, I think I just said, it's far more effective in modern countries which have ability to do it. If you look at what's happening in malaria in some parts of the world, people know it's close by, it's 100 kilometers away, may come across the border, they're monitoring. They want to know which mosquitoes they want to know what to do. Are we going to spray? Are we going to do whatever method we have to stop the entrance of malaria and malaria-infected mosquitoes into the area? Okay? Ebola is so unknown in that part of the world. Nobody was monitoring it. No one was expecting it, and it took everyone by surprise. Not if it had been in some remote village along a river of Zaire somewhere. 40 people would have died, would have made the news for about 5 minutes, and that would be it. The situation got into the urban situation, and there was no controlling it. And when the West did step in, it was pretty late, and it was panic mode. It was not unlike, some people have described it as what happened in the plague in 1300s. People panicked, left where they were going. There's a case in, I think it's Sierra Leone. The first case was a person from Sierra Leone who was from Liberia originally, went home to see his mother who was sick and his sister who was sick. They died of Ebola. He walked back, and he actually, they know places he stayed and places where he infected people with Ebola. If we know what we're looking for and we can monitor to try and figure out how it's changing, it's much easier. To sit and monitor for something which we have never seen in that area or have not seen in decades, it's really, really difficult to be able to invest those monies by a government for something which, in all likelihood, they're going to say is never going to show up. So can it work in developing countries? Absolutely. The government of Colombia invested a whole bunch of money on the Caribbean coast and the border with Venezuela for chikungunya because they knew it was coming. They knew it was coming. They just wanted to know where it was entering and what to do with it, because the pattern of movement was pretty obvious. With Ebola coming to West Africa, it wasn't obvious at all.
Yes, there.
So my question for you is, as climate changes over time, new species will start to emerge as they either die off or they're going to evolve and adapt to the new climate almost. So are these new species going to start being monitored for either immunities or new pathogens that will start coming in contact with humans?
If we know what they are, we'll monitor them. If we have next-gen sequencing data saying this is a brand new bacterium which can cause gastrointestinal pain or can cause liver cancer or can cause whatever, if we have knowledge of what it is, yes, we can monitor for it. We know that we're going to get new organisms. We don't know what system in our bodies are going to attack, respiratory virus, we don't know. Once we do know what they are, yes, we can monitor for them. But the trouble is trying to monitor something that we don't yet know exists I don't want to be fear-mongering, but there are things out there that we don't know. And I gave a talk a few years ago. I talked about the known knowns and the known knowns and the unknown knowns and the unknown unknowns of Mr. Rumsfeld, which we won't go into right now. But that is one of the unknown unknowns. That is one of the things we don't know. But we know it's there. But we don't know when it's coming out. We know it's there, so it's a known known, but we don't know when it's coming out or what form it's going to take.
Early detection. Sounds like monitoring and early detection are leitmotifs. Any other questions or comments? It's very warm in here. I don't know what's going on. I keep trying to open the door, but it's...
I thought it was me.
No, I know. No, it's usually not this warm in this room. So here, we have a question here.
Hi. Nice to meet you. I walked in here by accident. Your lecture...
So did I.
Your lecture was great. I come from a media and communications background. I don't study sciences, but I tell a story about sciences. It seems to me, and I don't know if this is the case or not, this is my question, is that there's a communication breakdown between, between one, government policies and funding departments. I'm wondering if this is the case. One analogy that you made was the missing puzzle, and that it seems to me like that may or may not be the case. So, is there a communication problem between those sources?
Yes, there are communication problems. Yes, there are communication misinterpretations. For simplistic forms, I can tell you that when a journalist interviews me, I say, do you mind if I take a look at your text before you publish it? No, I won't let you. I say, I'm not going to change what you want to say. I just want to make sure that the science is correct. And I have journalists with whom I work, and they say, absolutely, Carl, make sure I'm not saying something stupid. Okay? So there is that communication problem as well. We often don't speak the language, we scientists, of the politicians or of the media. Okay? And we have to learn to express ourselves succinctly for those 30-second sound bites, which people love to follow, but also clearly and cleanly exactly what we're trying to say. Don't blow it out of proportion, the reality, and try and communicate simplistically to make sure that we are not confusing an audience. We in academia tend to wax and wane politically using big words and big concepts because we do. We've got to realize that sometimes does not let us get our message across. We are not looked upon as any brighter because we can do that. We're looked upon as dumber because we can't express simple concepts to people. And so we have to work significantly with the politicians to speak their language so that they can understand what we're saying, and to speak with the press, with the journalists, all the rest of it, to make sure that they report what we are trying to say. And without a doubt, there's a big gap.
And you have to say things like early detection, not monitoring, because monitoring sounds too dull. Yes, sir.
I'd like to thank you for the lecture. I've learned quite a bit. I'm a nurse. I'm not particularly skilled in epidemiology or tropical diseases or anything like that, so I've found it quite informative. I do agree, I think there's a communication problem, but I think the basic communication problem, seems to me, is that power will not listen. I think the whole history, ever since before Kyoto, since Kyoto, and the whole trajectory that Western civilization is in right now shows that we live in a system whose economy depends on infinite growth in a finite system. And the political classes that run the system, which is based on a corporate mode of organization, simply will not challenge that modality. So we're confronted with a situation where, I mean, this is one of the aspects of climate change that we and our children will be needing to deal with, but it's only one of the manifestations. We're looking at massive population shifts, refugees on a scale never before probably seen in human history, wars over control of water and resources, increasing growth of national security states. We're looking at consequences we can only begin to grasp the nightmarish outlines of. And I think for me, the question is, what can science do to get the message to the population of the world as a whole that what seems to be happening is the debate is still very much open in terms of how rapidly and how far climate change is going to go. But that basically, it hardly seems controversial at all to say now the parameters are between the end of human civilization as we know it and the end of the human species. The consequences seem to be within 100 years, within sort of those two extremes. So I'm appreciative of all the information that you've given us tonight, and I would really love to see your presentation up on YouTube or available on the web to be able to actually study it a bit more closely. But I think that, speaking, I mean, this is just one aspect of a flood of consequences that I think we have to look at every possible way of finding an alternative to and organizing for an alternative to.
So thanks for that comment, because I think that actually highlights, we chose some of these topics because we think these might be the sort of wedge topics that can engage a broader discussion, so help with the communication. So disease is something people care about, food is something people care about. Climate change itself is just things are getting warmer, it's not such a big deal. Do you have any extra comment on that?
I thought I was the pessimistic one in the room, but you really beat me. Much of what you say is perfectly true. But the thing is, people don't really care. People don't care. Many people say, yeah, climate change, all the rest of it. It's a concept. Disease is real. Hunger is real. Having no water is real. So if we can get to people talking about climate change because of the effects that they are going to be feeling, I think we might be able to get their attention more broadly. In addition, in my classes, we talk about population density in the massive number of people in the world. So I say, how many of you agree, well, I'll ask this room right now, how many of you agree that we should reduce the world's population by 1/3? 1, 2, 3, you're gone. 1, 2, 3, you're gone. And they say, no, not me. Oh, not me, somebody else. And so everyone says, we think we have the solution, we have the understanding, but I don't want to be the one who has to suffer for it. Sometime down the road, we're all going to have to take responsibility and say, yeah, I'm going to have to do some suffering as well.
But your point, and the last 2 points about how that gets communicated by everybody, all different facets of society, is really an important one. I will draw your attention to the other topics that are coming up in the next 4 or 5 weeks, because we are going to talk about food, which should be another issue that people should care about. So we are going to talk about security, which came up. We are going to talk about environmental health, so some of this migration and how that affects our collective safety, and also about ethics. The talks will be up on a website, and they will be on YouTube, so I will post that next week when we get the website done. Are there any other, I don't want to cut it off. I'm cognizant of the fact that it is very warm, and I don't want anyone to faint. So, if anything else that people have to say? I hope, if not, I think we should thank Carl again for a very interesting talk.